Hi, Yashi Gayatri. Welcome to the podcast.
Hello,
So
Professor.
today we were going to take one step further than where we left in the podcast. Last podcast we did to tell you what sequences are. Why do we need sequence alignment? Anytime we do bioinformatics, people talk about alignment, so many methods for alignment. So we want to go to the real basic. What is alignment? What is the problem with alignment? Okay, before I start with that, I would like my co podcast people to introduce themselves. Yashi, you want to start?
Okay. Hello everyone. My name is Yashi. I have a master's in biotech, and I have worked in various projects, basically on wet lab and molecular biology. And my specialization is in genetic engineering. And I'm trying to build my concepts in bioinformatics now. Thank you.
Thank you, Yashi. Gayatri?
Hi, Professor. Hello, Yashi. Hello to everybody who's listening. I'm Gayatri. I have a master's by research in bioinformatics, and my work revolves mostly in gene expression, gene regulation, data analysis. And currently, I'm working to automate omics workflows for the industry. And it's happy to be here, and I'm looking forward to the discussion.
Okay, so I am Subhas Srinivasan. I have been teaching bioinformatics for nearly 15 years, and I've done bioinformatics research for 50 years. So it's 1550. Okay, so what I feel more recently is because of AML and all these onslaught where by clicking you get something so easy, you think you've done it, and the concept behind that is going to get lost. So I want this podcast to just hit at the bottom concept, not so much the methodology or protocol or anything, just at the concepts of everything. So we will start with why we need sequence alignment. We said it in the last podcast. Today we are going to say what is the problem with it, what are the challenges, and why we do the sequence alignment the way we do today. Okay. So, and where is the origin of sequence alignment? To do that, I will have Gayatri tell us a little bit about because she was very excited about this podcast when I said we are going to do dynamic programming algorithms. I want her to say what she was expecting from here.
Right. So, as students, whoever is studying biotechnology or bioinformatics in any capacity, we would have come across sequence homology, this concept of homologs, orthologs, paralogs, and the similarity between different sequences. It could be DNA sequences, it could be protein sequences. And we also use BLAST, FASTA, and all these algorithms on a daily basis, whoever's actively doing bioinformatics. Sometimes even biologists use them to have sequences, and then you just want to quickly see if a new protein or a new sequence is similar to something, you want to check it against a database. We do this on a daily basis, but how often do we think about how these came to be? And what is the origin of these algorithms that are so helpful to us every day, and where did these come from? Are they of biological origin? Have these algorithms been adapted from somewhere else? If they've been adapted from elsewhere, how did that problem translate to the sequences? So this is a very interesting part of scientific history, as well as like the maths and the concepts behind it. That is why it interested me a lot, and that's what I'm hoping to understand through this session too. How we do sequence alignment, what are the principles behind these?
Yashi, you showed much interest right on when I said we are going to be doing this podcast and all this. What made you too excited about this?
All right. So as I just told that I was trying to build my concepts from scratch in bioinformatics. So I think surfing on Google and trying to read research papers is like going down a rabbit hole. There's so much and everything, and it's very difficult for a beginner like me to understand from where to begin. So I am really looking forward to this discussion to to understand the very basics, as Gayatri just said, that from where it all began and how the layman knowledge was translated into computational biology. So I'm really excited to see that, what I can take from this session.
Thank you too. I feel the same thing is going to get lost if we didn't have this podcast. It's very basic. Yashi talked about basics, and Gayatri said history. I think basics are in the history. So we do need to go back in time to see who contributed to what. And sometimes names may not be that important, but in this case, we do know names. We will talk about it. So, how did sequence alignment come into being, and what it was telling? In the last podcast, we ended saying that for annotation of the proteins, we need to do that because similar sequences actually share similar function. Okay, we let it in the last one. But now, how do you use that similarity, similar sequences, similar function? How do you use that concept to predict the function of a given sequence? Okay. So I'm going to just start with Yashi here. Yashi has 100 amino acid protein sequences and she came and she want to know what the function of it is, let's say, right? And I have a small database of 100 protein. That's all I could collect in all my life. And I have annotated them and I kept it there. What she has to do is to compare her sequence with every one of these 100 to find out what is the best one and say perhaps the function of the 55th sequence in the database is similar to mine. Okay, that's still prediction, needs more validation, wet biology and all that. That you guys are learning a lot. Okay, but then that comparison I said, the comparison is such a big loaded word. Comparison means you're aligning. It's not comparing. You look at me and look at her. Yeah, they both have black eyes, the red hair, whatever it is, right? It's not that comparison. It's very complicated. And to compare two things, you need something, right? You need a cost element. Okay. So I'm going to tell you, two sequences that can be aligned this way, in between gaps, this way. Which one will you select? Because she has 100 amino acids and I have 100 in my database. So she has to do 100 by 100 comparison. Within each comparison, she has to explore all possibilities. What does it really mean, all possibilities? Then after all possibilities, how are you going to say which one is best? Right? For that, you need a score. Okay, you need a score. And we're going to come to that scoring matrix. Right now, if I just say scoring matrix, that is really pie in the sky. I don't understand. So let's quickly go back to a problem in businesses. Businesses, they do optimize a lot. They know how to optimize things because in businesses, it's the bottom line. They have to save cost. Okay, it's a major thing for them. For me, it's okay if the alignment took more than 3 days, it's okay. But it's not okay if it's going to take a whole year, right? So our costs are different. But the concept of cost can be time. The concept of cost can be computational time. The concept is you need to do it today. I want the answer today. So that kind of thing. So there are problems in businesses, in mathematics, where you need to find an optimal solution to save cost for a given problem. In our case, it's alignment problem. I will come to that, how the alignment and their problem become similar. In the case of traveling salesman problem, suppose there are 4 cities, A, B, C, D. Okay. And the condition is that salesman starts from home, of course. He has a home, he's going to get up in the morning and he's going to travel. Where does he go? His boss is telling, hey, don't spend too much money. Okay, so he has to optimize the cost. He has to visit all other 3 cities and come back home. Okay, he needs to be able to say which city he goes first, then next, and then next to back home. He needs something. Otherwise, how does he know? He needs either the distance between cities or he needs a cost to go from point A to point B. That cost is something important to optimize. So once he has the cost, how many possibilities? I said 4, right? I said 4. Is the air sounding too bad? Fan?
No.
No? Okay.
No, we can't hear him.
Okay. So the A, B, C, D, I will say that forget it, only 4 cities. I will just compute the cost and then start buying ticket. Okay. I can say A to B, B to C, C to D, D to A to D, A to C, D to C, D to B, back, and then diagonal. All possibilities, and the cost is there. I have a cost matrix. Okay. So that cost matrix, I keep adding and say, I got it. Okay, first go to C, then you go to B, and then I go to come to D and A back. Okay, I got it. That's optimization. Just for 4, I can do it by hand. All possibilities. Sometimes you can have 30 or 40 cities. Everything has a cost, and you have to optimize. So as in businesses, the optimization problem gets crazier and crazier because there is not only one. It's not just a cost. Okay, it's also someone may want at 12 o'clock, some other person only want to at 3 o'clock. You have to, that also optimize. So too many parameters, but let's just worry about the cost at this point. So I have 20 cities to go and I need to compute. So 20 cities, all possible. I don't think I'm still patient. I'm from the 50s and 60s. So I don't think these days you will do it by hand, right? Oh, forget it, ma'am. I don't want to compute all the possibilities. Is there a solution? And then I'll say, yeah, there is a solution. That is dynamic programming algorithm. They use it. That will tell you in one stretch what is the most optimal path. That was existing in traveler's salesman problem, in matrix multiplication, okay, where you clearly know cost from point A to point B, okay, and then you want to know where you're going to go, which point are you going to go, how are you going to travel, okay. So those two are almost like I'm giving you as a prompt. You can go into ChatGPT or YouTube or something. There's enough. They tell you how dynamic programming algorithm applied to solve this problem. This multi-dimension problem into a clean n-square problem. Okay, so 20 cities, 20 by 20 computation. 100 amino acid, 100 by 100 computation. Okay, so it is not 100 times 100 times 100, n cube. That will be the case if we did not have the cost. Yeah? So, Gayatri, tell me something about the cost in amino acid mutation. Have you heard anything about that? Do you know? Yeah.
Yeah. So we see a lot of diversity in proteins, right? So the possibility is that there are 20 amino acids, and these make up for a lot of different combinations of proteins that are possible. And this in turn comes from possibilities in the DNA, the codons. So mutations in proteins would be a very important thing to consider in this approach, because like you said, the cost of having a different amino acid at a given position and the cost of just not having any amino acid at that position, because if we were comparing two sequences, a longer sequence and a shorter sequence, right, how would you know where at what positions is this sequence similar to that, and so on. So that is a natural cost that we are trying to calculate computationally. So I believe that would be a challenge, a computational challenge when doing dynamic programming, to see what is the natural cost of one amino acid being substituted by the other. But then the computer would not know this. This should have to come from the underlying biological information, and I can imagine how difficult it would have been to come up with this cost in the first place. So yeah.
Vyashi, you want to say something here? Quick, what are you looking forward to hearing now?
Okay, so I would like to understand that what kind of cost or what kind of penalty one has to, apply when we're talking about alignment. Yeah.
Okay, thank you, Yashi and Gayatri. So I'll take to that. So the cost or penalty, she said. Penalty is a nice word because cost is also in the opposite way. It's penalty only, right? It's going to cost you more. That's a penalty. So you want to walk the path. That's another word Yashi just introduced. That keep in mind because people will use different words. Okay, they mean the same thing. Yeah. So Gayatri was saying, how do we even come up with this? What is the cost of mutating A into a T? So for example, two sequences are very similar except in one position where alanine becomes a threonine. Okay, so I'm saying that, okay, what is the cost of going from this sequence to this sequence where only A has become a T? Okay, so A to T substitution, okay, has a cost in nature. Okay, it can sometimes ruin the protein. We will never see those proteins. Okay, so those are all kind of silent witness, they call it. We will never see them. Okay, but you see what you see A to T, both of them got accepted in nature, meaning that both are making the protein and it's still functioning in some organism somewhere. Okay, these are called accepted mutation. How often does it get accepted? That is something that had to come. We have to give credit to Margaret Dehoff. Okay. I'm equally happy that she happens to be a woman. Sorry, I'm not. You could call me a feminist, but I get excited whenever women scientists at the time when things were so difficult. They were so anxious to come up with really breaking science that really changed the course of molecular biology, changed the course of how we do genomics. She was a woman and she was not a computational biologist. Okay, she is a biochemist. And her question was, she takes, suppose she had the lysosome from lysosome from human, and then she got the homologue from mouse, rat, whatever farm animal she had in her backyard, right? Those days, she will go and get it from the blood, and she sequenced the whole thing, and she saw that they were all not identical. They were similar, but in places they have gotten mutation. Why? Because I told you in the last podcast, replication is not perfect, but replication error doesn't always have to translate into amino acid error, but rarely it does. When it does, then nature said that it's okay, I will tolerate it. Okay, so that is the toleration is what we are calling as cost. Once a particular amino acid can go into another amino acid more often, okay, then the cost is low. Cost is low means score is high. Okay, cost and score goes opposite here. Okay, the score is high. You're saying it's more often. Score is high. It cost means doesn't happen. Just the opposite, right? And so if I say that alanine is, I say, is mutated by aspartic acid, then I have not seen it that many times, but it has happened in few cases. Then I will score it lower. The cost is a little higher. Okay. Every time I'm bringing score and cost because in businesses it's cost. Higher cost, you don't take it. In alignment, lower score, you don't take it. That means it's less probable. Okay. So the whole thing is about probability. And why did Margaret Dehoff, I'm just falling in love with her head over heel, is because she was amazing. When she sequenced all that, she did align them by hand. 1,000 or I think 1,100 some amino acid sequences she had in a database. Those database was not there in a computer hard drive. I'll come to this hard drive part, okay? But she had them all printed, okay? Because she can keep only one sequence on the screen or something. I don't know what kind of a computation problem she had, but she had printed all the protein sequences on a book, which is called Atlas of Proteins. Okay, so and then she aligned every family. Suppose in that there were 10 or 15 families of proteins, serine proteases, and phosphatases and all that. She aligned all the serine proteases into one multiple sequence alignment. How does she know all of them are serine proteases before aligning? Because she is the one who did the biochemistry. Okay, so she knows they're all same. So she dared to align them. Today we cannot do that unless two sequences are you know that they're similar. You can't even align them. So she did the multiple sequence alignment. This is also a prelude to multiple sequence alignment, which are going to come later. She knew the functions are similar. She's the one who did the biochemistry. So she aligned them. And she said, oh, more often I see alanine going into leucine. And those alignments, she did it by hand, I have to tell you that. Why? Why she did it by hand? Because there is no scoring matrix for her. She needed scoring matrix to align it, so she did it by hand. Her I was the scoring matrix. Okay. And computers don't have I, okay? So we need a scoring matrix. Okay. So she did it by hand, and she found out in those 11 or 12 multiple sequence alignment coming from 11 or 12 different protein families. She saw how many times alanine went to threonine, how many times leucine went to isoleucine. And based on that, the more times leucine went to isoleucine, she said that's more likely, more acceptable. So I'm going to score it high. Here I must mention, if something is not mutated, that gets the highest score. Because alanine to alanine, there is no mutation, highest score. Same here in the traveler salesman thing. If he is not traveling at all, the cost is 0. Right? But that's not what we want. Correct? So we want the best optimal, right? So that's how she came up with a PAM1 matrix. Okay, where she said, I have a matrix now. Now, as more and more sequences come, I don't have to keep aligning them by hand. And also, I can dare to take more and more divergent sequences and still say that there's a functioning acidine protein. Still say that. So she started the concept of mutation matrices, from which we have now, Gayatri will tell you about Blossom and everything that she uses, right? Yeah, tell me something about it. Like you use the Blossom matrix a lot, right? Yeah.
So yeah, first of all, yeah, I agree with you that it's nice that it's a woman, and we would have to say the mother of maybe sequence alignment or something instead of father for a change. It's a great thing. So yeah, this brings up a lot of learning memories, because we start with this PAM and Blossom matrices, the point accepted mutation matrix, and so in this PAM matrix, what she essentially was trying to do is to basically take a note of how mutations are happening over this, and then over a lot of sequences, and then later they had to because they were not sure about how these mutations were progressing, they had to move on. They were not confident about this. They had to move on to block, which is Blossom block substitution matrices, where they went block by block. And then I also know that this matrix had to be multiplied multiple times. So can you tell us about the PAM 250 that we hear about, and Blossom 62 and then?
Yeah. So because she took sequences that mostly had one mutation per 100 amino acids, so she called it like one time mutation. So then she said she wanted to go to include those proteins that are not that similar. There are more 3 or 4 mutations between 100 amino acid because they're distant. Then maybe 50 mutations for 100 amino acid. So how do you come up with it? She said, just don't worry about it. Just multiply the PAM1 matrix that many times to handle more divergent protein. When it comes to divergent protein, you can't even start aligning, because then you will get gaps. Even if gaps comes where there are sequence lengths are changing, then it's already too complicated. To just do it by hand. Even if you wanted to do, you cannot do it. Even if you have patients of very patient from the 50s and 60s, you still can do it. It's impossible. Okay. So Blossom, as Gayatri was mentioning, Blossom is, of course they have to use PAM now. Okay, PAM 100, PAM 250. They use PAM 250 to come up and align all the protein that we have now. By, I think Blossom was done in 1991 or something, around that period, 15 years after PAM matrices came up. So they used PAM matrices to come up with blocks of where more confident multiple sequence alignments are there. From within the blocks, then they again computed what are the amino acids that are most likely to change. Same way as they have did. Now, instead of taking the full sequence, they had blocks. So that way now blossom is used more often. Okay, I'm going to tell you something. Blossom 62. What does it mean blossom 62? That means blossom 62 came from all the proteins that have identity identity higher than 62 amino acids out of 100, 62%. So that
in
number tells
PAM,
you how
sorry.
divergent. Yeah, go ahead. Go ahead.
Sorry. So, but in PAM, it would be in the number of mutations per a set of sequences,
Yeah.
right?
Yeah.
Yeah. Okay.
So here also they said, okay, if I'm going to take all the proteins that are within 62% identical and then get the blocks, then make the matrix, that is blossom 62. If I took all the proteins that are 50% changing between each other, identity, I'm saying when 100 amino acid means 50% of the position they're identical, other 50 position they're changing. That's what I mean, Yashi. Okay. So if I take that and come up with a matrix, then that's blossom 50 and so on like that. Right? So anyway, so that's how the blossom matrices come. So
Can I say
so the
something quickly?
numbers have, I do want to, I may have misrepresented 62 and 50, but this is the podcast, so you guys can go and look. Okay. So that really means how sensitive is a matrix to sequence alignment. Okay. How much you can trust and how much you cannot trust. And that's the point here. So if you don't know the basics of where is the 62 coming from, where is the 50 coming from, where is the 30 coming from, where is the 250 coming from, then you will use it for something else that's not even applicable. The sensitivity of these matrices drop below that. So just to know where they're coming from, they all have limitation. Okay. Now we have a matrix. So are you happy with it, Yashi? Are you happy you have a matrix now? Okay. So what does the matrix mean? Matrix now for protein matrices, it will be a 20 by 20 matrix, right? Because only 20 amino acids 19 amino acids can replace a given amino acid. Right? So you need a cost of alanine going into arginine, alanine going to leucine, alanine going to isoleucine, and same way leucine going into alanine, leucine. So it will be a 20 by 20 matrix. And every element has a cost of that. And where did the cost come from? It's a cost come from probability of amino acid getting changed. It's not cost here, it's a score. Means it's going to get more marks like your examination mark. More mark, the better, right? So that's how the mutation. The probability of an amino acid going into another amino acid by looking at many multiple sequence alignment, you say that, okay, the least ones are more unlikely to happen, so they score very low. Okay, so that's how those are computed. So for math also, you can look into some lectures that are there, very well defined lectures. How did PAM matrix actually compute? How do they compute? So, but for here, it's enough to know that they are actually cost or score. Okay, so now we have a score. Okay, now we got a score. Oh my God, it took us so much time to get the score. Now, how are we going to align? Okay, that is where we are looking back into the dynamic programming algorithm. There is this guy who was actually Gayatri when I said that next podcast will talk about dynamic programming algorithm. She was how did this happen? How do they know that will work and stuff like that, right? Like I'm equally excited that how do the people even do that? So there was this guy perhaps, okay. His name is there somewhere. I don't remember off the top. He was
Richard Bellman, I believe.
I say that again?
Richard Bellman.
Okay, Richard Bellman. So he's the one who found dynamic programming algorithm. But there is another guy I'm talking about who was sitting in the God's balcony and he's saying that, okay, these kids are playing around here with dynamic programming algorithm to solve business problems. Yeah, that's good. They're playing very well. They're not making their hand dirty and doing good. Okay. And then he looks around here because he's sitting in God's balcony. Sits, oh my God, this molecular biologists, David, Margaret Dayhoff, including, they're all struggling to align sequences. And Dayhoff has come up with a scoring matrix. Still, they're struggling with aligning sequences. They could just look in here. And his brainchild is to say that dynamic programming algorithm that's applied to traveler salesman problem, matrix multiplication problem to bring efficiency is exactly the same solution that's needed for sequence alignment. Yeah, because you have the same scoring matrix, you can do the same thing, min max, so you don't have to go through n cube or even more number of operations to get that solution. And here, I'm going to add a little bit in sequence alignment. The 20 by 20 is not sometimes good enough because sequences change in size, right? Like maybe Gayatri can tell because she always brings up the point like shorter one, longer one. So tell us all like what do you think of these gaps? Where are they coming from?
Yeah. So like if we talked about sequence alignment, if you're approaching it as a problem, let's say we're trying to align two sequences, it is well and good as long as we have two sequences of equal length, let's say 10 letters. So it's easier that way. We can align them just by matching them with our eyes. Probably that's what Margaret Dayhoff also did. But imagine you're dealing with like a full protein and just a domain, or let's say just a string of like 8 letters and then 5 letters. The problem comes where when you have to align these two, the goal always here is to maximize the similarity, right? To see where the 5 letters of the second sequence fit on the 8 letters of the first sequence, or whatever the sequence length is. So that could be anywhere. These 5 could go anywhere. They could go for 2 at the starting and 3 at the end, and the rest is a gap. Or these could go alternate. All the 5 letters could go alternate, but there's no way that the computer would know that, right? And this also has to make sense biologically, and that would also be a challenge in computation to understand where the gap should come while aligning these two. So that would be a problem. I'm assuming where the gap should fit, how many gaps should come, and how should we penalize these gaps?
That's where the penalty comes also, right? As Yashi was mentioning, how do you penalize it? So, Yashi, you getting the point on the gap thing? Yeah?
Yes.
Yeah. One other way I can tell gaps can be understood is this traveling salesman problem. Suppose he has to go those cities, but right between when he was traveling, he gets a call from his father that his father is sick. So he has to touch base with the fifth city. Okay. But that makes it efficient because if we didn't go to the fifth city and he just travel the cities as the boss said, his mood is not there. So he's not doing his job well. So he needs to give a gap. Okay. To cost management is not always just doing everything exactly by the routine. Sometimes you have to add gaps to make it more efficient. So this is just a cockamamie way of explaining where the gaps can come from. In biology, that's how the gaps could have come. Just remove the three bases completely randomly. 3 bases went away. Then that creates one amino acid gap in the protein. But the nature has accepted it because the total cost was less. Okay, so that way now you have to see where did it accept it because you don't know the sequences. Sometimes sequences diverge too far. So gap poses a major problem, yeah, because you don't know the gap. I said that okay, there is no gap. I can go from myself, keep on aligning, shifting one by one. Computers don't have eyes. I have to say it again and again and again because otherwise we won't know what the problem is. They don't have eyes. They need the scoring matrix to tell even A against A is identical. Okay, I like it. It can't even see A is identical to A. It has to look at the scoring matrix, right? So that's a problem with the computer, but they can scale very well. That's where we can say, okay, you do this. You do dynamic programming, you just scale. You fill all the matrix element and do all that. I'll sit. I know that I have the method, I have the scoring matrix. I'm just going to go for a coffee, right? But with the gaps, the problem with the dynamic programming algorithm is so much more efficient. Is it knows it can optimize where the gap also goes. Otherwise, gap can be in the first position, second position, third position, fourth position. You have to complete all options. Then, if I say there are two gaps here, as she was saying, there are three gaps in your sequence, right? Yeah, Gayatri. 8 and 5, three gaps. One here, two here, one here, one here, one here, or three here, none here, or the three goes at the end. Too many possibilities. You cannot, even though it's 8 amino acid and 5 amino acid, forget it. All possibility. If you have to compute by hand, I wouldn't do that. Okay. So that's why dynamic program. And the gap penalty it is not coming based on evolutionary thing, because that's hard to come up with. So we just say -2. Okay. -2 is the gap penalty that seemed to work. It's a practical, empirical number we came up with because it seems to work. Because when you know this is the right alignment, when you put the gap penalty -2, it does a decent job. Okay. So that's where the gap penalty comes from. And so, okay. So you want to, Yashi, you want to say how far you gotten with your concept? There may still be gap. I'm telling you, there will still be gap. Okay. But there are some prompts, whatever you heard, you can now go and understand. What will you do now after all this information?
Okay. All right. So from the conversation that, we just had, I understood that while aligning different amino acids, we can witness gaps. Okay. And there are certain gaps which are already accepted because it does not change the overall function of the protein, right? Just because if an amino acid is changed. So, but since we are aligning the different, the sequences, we need to score them. So the score of -2 is assigned as a gap penalty that we can say. And yeah, that is all I would like to say.
Okay. So the gap penalty, the gap can go anywhere. So there will be A to gap, L to gap, every amino acid to gap one. So those all go into the dynamic programming algorithm. I know Gayatri was explaining how they are taught dynamic programming algorithm. Can you go ahead and say how you fill the matrix and what do you do? What did you understand from that?
Yeah, so I remember the first thing, first we are taught the concept and then we start filling the table, basically this very famous table where we try to align two sequences. Then so let's say there are sequences of 10 by 10, just for the sake of ease. We leave one line for the gap and then we start filling. Usually that is where our curiosity ends. Once we figure out how to fill the table, we're like, okay, I got dynamic programming. But so we progressively increase the gap penalty while filling it, and then at each score, basically at each cell, basically what we are trying to say is so we're basically trying to match the residues, right? So we are trying to understand the effect of the previous residue on this residue. That is what essentially, in my understanding, dynamic programming is, where we break a big problem into smaller problems. And then at every step, we're trying to find the best way to reach that residue from the previous residues. And then we save the score so that we don't have to compute all the way again. Because otherwise, if you're doing like the whole thing, we have to find out all possibilities. How did we get from A to L and so on, like at every step. But okay, so we filled one and we get the best, the score. And then by the time we reach the next one, we are considering the previous one, the one that's below it, the one that's above it. And then we start filling based on the calculation that we have. We have like a score where we take the maximum of the three values, the one on top, one on left, and the one diagonal to it. By the end of the matrix, we reach a maximum score. So that is considered the perfect score for this alignment. And then we do a very important step called traceback, where it's like back propagation that we do. We go from the last score and say, how did we arrive at this score? We check the maximum score at each stage and then reach the start, and that would have been the score for the alignment, and we would have aligned it.
So that's great. Yeah, she probably hasn't done this. Okay. So there are places in on the web you can see how the matrix are filled, what she was talking about. But I'll tell you one thing which may be missing there. You said that okay. So first four elements. Okay, 1 1, 1 2, 2 1, 2 2. Okay, the 2 2 element you are filling. Okay, 2 2 element when you're filling and you're going down. Okay, the 2 2 element when you're filling, you take values from here, from the left, from diagonal up, from the top. Between the three values, you're saying that which one is the highest, I'm going to put it there. And I'm also going to remember the highest came from left, top, or from the diagonal. Because that will be useful while tracing back. Got it? Okay. Why these three? Concept, I'm just going to add a little bit concept, Gayatri, you would love it. Why left, diagonal, and top? What does it really mean? Okay,
I'm not sure.
I'm going to tell you. Okay, when you're taking the value from diagonal, you are saying that I'm not going to give a gap. I'm going to substitute. I'm going to put the next amino acid in row and say how much is the value. When I'm taking from the left, I'm saying that I'm giving a gap on this sequence, gap that is on the column.
Right.
When I'm taking from the top, I'm saying that I'm going to insert a gap on this sequence, which is on the row.
Right.
Any position in the alignment, there are only three things that can,
Possibilities.
either it is matching or there is a gap in this sequence, a gap in the other sequence. Those are the three you're filling, and that is the concept.
Right.
But since you're picking the maximum, you're saying that suppose it came from the diagonal, you're saying that more likely there is no gap between those two. If you fill the next element from the left, there's a gap between this and that. Gap in this sequence compared to here. Okay, so all of us have to understand it in our own ways. But the three step that's going to come all the way into doing HMM model. Because there are only three states. A given, you were just telling me that you can break it into smaller problems. So this is the smallest you can break it to. You're saying that one position can accept another amino acid, or you can have a gap here, or you can have a gap on the other sequence. Gap can be on the first sequence, gap can be on the second sequence, or there's no gap. Only three choices. That is how you broke it down into smaller and smaller. Then you're walking directly from there, and that's how it's coming. Do you like it?
Yeah, that actually makes a lot of sense. And also clears the uncertainties. Like, can I just randomly put numbers and come to a solution? But at every given point, we're accepting only three solutions, and we are mapping from there.
Yeah. And you're taking the maximum at that point. So you made the decision at that point itself. But while pulling also, you're pulling the exact way where the maximum solutions came. That's why you have the arrows, right? Yeah?
Yeah.
So there are only every position, 100 position, there are only three states. You're evaluating every time. And I want to come to Yashi a little bit. I don't want her to be lost. Okay, go ahead. Tell me, did you get it? Because she didn't know how the matrix is filled. She hasn't done that. So this is a good thing because now you'll go and do it. Okay. So when you're filling the matrix, okay, each element, did you get the concept that there are only three solutions?
Yeah.
Because you're aligning a big sequence, but every position, there are only three solutions. Either a gap goes here, a gap goes there, or there is no gap.
Okay.
You got that, right?
Yes.
Now, when you go and do the dynamic program, you can actually take a short lesson there. You will know what's going on.
Sure.
I'll add on one more thing. You just mentioned Gayatri because most of them just go by the protocol, right? This is the most beautiful thing in dynamic program diagram. On the top, you said you fill the all gaps, right? -2 plus -2 plus -2 plus keeps going like that. On the left, you have -2, -2, -2, -2, -2. And then you start filling the matrix. Because you need something in the top, something on the left, and diagonal starts, right? First element. Okay. So what does the top really mean? The end of the top, the score at the end of the top row. That score is like you starting to align the sequences this way. Everything is a gap here. Okay. Okay.
Yeah.
And the column all zero tells you everything is a gap here this way. That is how you're starting. And you're slowly starting to move them. Tuck, 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 tuck. Got
And
it?
I also remember something, I'm just remembering from what you're saying. Is it right that if there are two gaps, one after the other, that is less likely to occur? Is there
It
something
is
that is
more
also
it's
taken into
more likely to
consideration?
occur. No, it's more likely.
More
to
like
occur. Why? Because again, see, when we talk about mutation matrix and alignment and everything, we're forgetting a little bit of biology, right? In proteins fold. Without folding, there is no function. Okay.
Yeah.
So anything that happened to that protein from human to mouse or human to cow, they're all accepted. Okay. So proteins have a core, right? It has a core. Core, any amino acid change and deletion, insertion are no in the core region. Because then it won't fold, then it won't function, then that sequence won't even exist. Since it is existing, we are saying that now we know it cannot be in the core, it can be in the loops. Still it can fold because it comes like this and it's going to fold. Near the fold, maybe it's a little shorter fold, longer fold. Then you can accept that. So from the structure point of view, some regions of the protein can accept more mutation, more gaps. So there is a affine gap penalty where you say that first gap insertion penalty is much more. Then you say, if I have already inserted it, then the next gap is more likely to happen. Then I will penalize it lower. I'll say first gap -2. Next gap, if it follows it, it's -1. I think I'm going to tolerate because in my mind, I know it is in the loop region, perhaps. I'm going to protein, although the computer doesn't know anything. So we are really building a little bit of knowledge base in all these matrices. We know exactly what happens in the protein. That's why these penalties are also, we are softening it to actually understand the biological problem. Okay, so now how we are going to fill the matrix, we are not going to say in this one, because that I think is being taught in the classes throughout wherever, whoever is teaching dynamic, and it's fairly straightforward method. There's a min of this, you just compute it. So we won't do that here. But the concepts, I think we have covered everything, Gayatri and Yashi. Do you guys get it? Yeah? Did I miss anything?
No, I think to quickly summarize what we've gone through, we've started
can
with
you do that? Do that. Yeah.
Yeah. So we've started with the need for alignment and why there is a need for alignment and why that is a problem in itself, why that should be a problem, because obviously there's a lot of sequences and how it was approached in the start. So we've discussed Margaret Dayhoff, how did she come up with PAM? She had to do all that manually, and so once we got, so this was the basis. First we needed a scoring matrix, so how we came from sequences to a scoring matrix, and then after we have the scoring matrix, now we have unknown sequences, a lot of sequences, so how we scored them, why is dynamic programming coming here, and how dynamic programming approaches the problem. We also got a nice analogy for gap from the traveling salesman problem. And we also got this very interesting chicken and egg thing where she had to align to score, and then we use the score to align it now. So yeah, I think we've covered a lot of the concept from the start to what is currently accepted, I feel.
Thank you, Gayatri. Yashi, tell me.
Okay, so I really liked to, that I was able to understand that, why we are actually doing it, that why we are actually talking about gap and why we are actually talking about a penalty. And instead of how, because we can really, as as ma'am said, that we can definitely learn it from classrooms or from YouTube videos, but to understand that, why it's happening, to solve that question, I think it's really core to, for me at least, to understand. So we started from, as Gayatri just said, that we started from understanding that, what is alignment, why we need it. And then we discussed about Margaret Dayhoff and her work in giving us the PAM metrics. And then we discussed about, the scoring methodology. So yeah, I think that's a very good start for me to,
So,
And
know more
Yashi,
about this
yeah.
further. Yeah.
Yashi, what I would really like for you to do, since you really wanted to learn, I want you now, because Gayatri did it the opposite. She learned how to do the dynamic alignment using dynamic program algorithm, and she's coming back to concept. And then, since you got the concept first, you haven't done dynamic program. Now, if you go and actually take a small exercise and follow the dynamic programming algorithm from other public sources, how to do it. Our next podcast, whatever the topic is, I will let you start with. Was it fun, like knowing the concept and doing it? And and then both of you can compare and say that, oh my God, I was just doing it blindly. And now I know the concept, why it's happening. I understand better. It was fun. Okay. In any way, concepts are fun because once you know the concept, you never forget it. Like I always tell students, if you wake me in the middle of the night and ask me what is the area of a circle, I'll spit it. I don't need to remember it, right? It's there deep. So concept takes matters deeper into your brain. So you forget, and because you put deeper in the mind, that concept gets connected with some other concept that you will do long time after in your life. Oh my God, it's the same thing they were doing there. And maybe you will invent a new connection, just like I told you that about the man in the God's balcony. You could be sitting in the God's balcony because you understood this concept long after I am gone, long after many. You can say, oh my God, I heard this long time ago. I did this podcast. I know that applies here. That's what happened. So anytime you understand the concept, they stay. They stay with you, they travel with you, they help you to solve other problems using the same concept. Yeah. Thank you both for coming here. Very encouraged. I'm really happy. I think we didn't go for much, so I think this is a good recording. Let me stop recording now. Record setting. Wait. It doesn't say. Oh, okay. Here I have.